Ekis. Welcome everyone to another episode of SCSC 681 Soil and Crop Sciences Seminar, the greatest show on earth. At least that's what I've been told. Today we've got a terrific trio of top-notch students presenting their research for your informational, educational, and edificational needs. Yes, this is the one-stop shop for all things soil and croppy. Full program today. Let's not waste a lot of time. Dr. Howe, would you like to introduce our first speaker?
I am happy to. I appreciate that wonderful and entertaining introduction as usual. So today it's my pleasure to present Samanta Hostetler. She is from Virginia, and she is doing, she did her BS in environmental biology from Christopher Newport University in Virginia. And she's doing a master's non-thesis. And after this, she's going to head off to George Mason University to do her master's in public administration in environmental science and public policy. So I want to welcome y'all, and she's going to do a very non-traditional seminar for you today. Take it over, Samantha.
Thank you, Dr. Howe. I'll go ahead and share my screen here. Okay, and everybody can see that okay, I hope.
Yeah, we can see it.
Okay, great. All right, welcome, howdy. Hope you guys are having a good afternoon. Today, I'm going to be talking to you guys about promoting soil health through research, extension, and education. So like Dr. Howe said, I am a master's non-thesis student. So I have a big project that I work on for my degree requirements. And that project is, I refer to it as the Soil Health Video Project. And it's a series of 4 different videos, and I'm going to be sharing 2 of those with you guys today. Both of them are in the drafting stages. So if you have any feedback or anything like that about the content or anything, feel free to email me afterwards, or you can bring it up in the questions after as well. Definitely willing to hear any constructive feedback on that. The 4 videos I am doing are, the first one is Introduction to Soil Security and Regenerative Agriculture. Video 2 is Nutrient Cycling with the focus on nitrogen. And then video 3 is Importance of Carbon and Organic Matter. And then video 4 is a bit of a closer look at some regenerative agricultural practices, specifically flash grazing in cover crops. I'm also going to talk to you a little bit about a small research study that I did in the lab today as well, which is a plant residue incubation study. And we'll talk a little bit about some of that data and maybe some opportunities for further research in that area. But we're going to go ahead and get started with the videos. So the purpose of the soil health project that I'm working on is to introduce the topics of soil security and regenerative agriculture within the context of nitrogen cycling, carbon cycling, and organic matter as it pertains to soil health. Each video is meant to dive into these topics in a way that is easily digestible to many different audiences. So the goal is to make it easy to follow, easy to understand, and sort of help get this information out in a way that the general public can digest. The final video in the series on flash grazing is also meant to be a closer look at one specific regenerative agricultural practice. But yeah, the purpose is to aid in education, extension, and outreach. So hopefully hopefully when you guys watch them, you feel that it does that. If not, I'm totally interested in hearing comments and feedback. So with that, I'm going to show you guys the first video, and then we'll hop into the second one.
Sustainable agricultural production relies on the soil's ability to provide support, water, air, and nutrients to plants, which is commonly referred to as soil health. The concepts of soil security and regenerative agriculture are also used to indicate efforts to restore or maintain soil health for sustainable production. Here, we are going to introduce these concepts and direct you to other videos in this series to learn about the science behind a healthy agricultural soil and practices that promote it. Before we get into that, let's break down what soil security and regenerative agriculture actually mean. Soil security has been defined as the ability for soil to sustain functions to provide planetary services and human well-being. Soils perform essential functions such as supporting plant and animal growth, regulating water supplies, recycling wastes, and serve as a foundation for roads and buildings. A house needs a strong foundation to keep it from falling into disrepair, much like we need strong and healthy soils to keep us thriving. When we focus on conserving and protecting the soils, we will also find that soil security can help solve other global problems involving energy security, food security, water security, human health, and biodiversity. So how do we go about improving and protecting soil health then, if it can help with all of these problems? Well, one way is through the use of regenerative agriculture, which is defined as the process of using different agricultural practices to enhance the soil. Some examples of regenerative agricultural practices are the use of crop rotation, animal integration, and managed grazing. In addition to these examples, the use of biological amendments like compost and manure, cover crops, and rotational grazing are also common management practices. So how do these practices enhance the soil? They can help improve nutrient cycling, rebuild soil organic matter, restore soil biodiversity, and improve soil structure in addition to numerous other benefits, some of which include decreasing soil erosion, encouraging water infiltration, and improving water quality. If you want to learn about the science behind these practices, please watch the following videos in this small series as they each talk about nutrient cycling, soil carbon, and soil structure, and how these characteristics of soil can be improved through the use of regenerative agriculture. On that note, thank you for your time and attention, and I hope I piked your...
Sorry, it cut off a little early. So yeah, this video leads into a couple other ones like I mentioned. So I'm going to go ahead and show you guys the second one. And this one's nutrient cycling, specifically looking at nitrogen.
The nutrient cycle describes how nutrients move from the physical environment into living organisms and subsequently are recycled back into the physical environment. This movement of nutrients, essential for life, from the environments into plants and animals and back again, is a vital function of the ecology of any region. While there are a number of important nutrients that cycle through the environment, the one we are going to look at more closely is nitrogen. Nitrogen is considered incredibly important because it is needed for the construction of amino acids and proteins, which is an essential building block of life for all organisms. It is comparable to oxygen in terms of biological importance. However, unlike oxygen, nitrogen in its natural atmospheric state is not usable for most plants or animals due to a strong triple bond between two nitrogen atoms. This means it must change its chemical form before it can be used. This series of transformations of nitrogen in the environment is referred to as the nitrogen cycle. In order to best describe how nitrogen cycles through the environment, let's start with atmospheric nitrogen. Nitrogen gas in the atmosphere can be converted by select microorganisms to form ammonia and then ammonium, which is biologically available. This is called nitrogen fixation. Nitrogen fixation can also be carried out by a human-driven process known as the Haber-Bosch process, which requires considerable energy in order to break that triple bond. Once formed, it can be released to the soil where it is available to plants and other organisms to use. In the soil, ammonium can be transformed to nitrate by two specific bacteria, Nitrosomonas and Nitrobacter, in a process called nitrification. Ammonium produced by the Haber-Bosch process can also be converted to nitrate through industrial chemical processes. Both ammonium and nitrate are biologically accessible forms of nitrogen in the soil, meaning plants and microorganisms are able to use both nitrogen forms and incorporate them into their tissues. This is important because humans and animals acquire nitrogen typically in the form of protein in their diet. Assimilation of nitrogen leads to a large amount of organic nitrogen in organisms that can then be released as waste or in the form of dead organisms or tissues like leaves. The green color of plants is actually due to a compound that contains nitrogen. At this point, a wide range of soil organisms will start to break down the residues. There are many roles and organisms involved, but fungi and bacteria are often the main decomposer organisms in soil. Decomposition converts the organic nitrogen to inorganic nitrogen, which is then released back into the soil as ammonium. This process is called ammonification. The ammonium can then either be used by another organism or undergo nitrification and then be utilized via the assimilation processes previously mentioned. Nitrate can also be converted back into a gas and lost from the soil through a process called denitrification. This process typically occurs when oxygen concentrations in the soil are very low. This most commonly occurs in soils that are excessively wet or flooded. In these conditions, anaerobic organisms convert nitrate into gaseous forms of nitrogen. Depending on the situation, there are three major gases produced: nitrous oxide, nitric oxide, and nitrogen gas. Both nitrous and nitrous oxide are greenhouse gases, while nitrogen gas is the final link to return the original form of nitrogen in the atmosphere. In addition to these processes, there are a few other minor processes in the nitrogen cycle. For example, lightning can convert nitrogen gases into nitrate, which can fall with rainwater. Under high pH conditions, ammonium can convert to ammonia gas. This process is very common in areas where manure is stored or accumulates and contributes to odor issues. So we've come full circle through the nitrogen cycle. Plants typically take up ammonium and nitrate from nitrogen provided by or recycled from other organisms through the nitrogen cycle or directly from compounds generated from the Haber-Bosch process. The Haber-Bosch process revolutionized agriculture by boosting yields. Unfortunately, it was initially very cheap and often too much nitrogen was added to the soil. When over-applied, nitrogen can run off the land, entering waterways. In the water, it stimulates plant and algae growth. Eventually, they die, which stimulates microbial decomposition. These organisms consume oxygen and lower water pH, which kills fish and shellfish, resulting in dead zones. An example of this occurs where the Mississippi River empties into the Gulf of Mexico. Regenerative agricultural practices seek to improve the entire agricultural ecosystem in order to rejuvenate or enhance all natural cycling processes and help reverse climate change. This includes enhancing soil organic matter, which improves recycling of nutrients, conservation of water, and reducing erosion. A variety of practices may be used alone or with others. Practices include crop rotation, reduced tillage, maintaining soil cover, residue management, and integrated farming. Prior to the Haber-Bosch process, integrated farming was commonly practiced. Farms were small and typically required animal labor, thus it was easy to rotate pastures and grazing with row cropping. As farms became mechanized and fertilizers more plentiful, animal and crop production separated. Intensive pressure of a growing population for food, competition for water and energy, and negative impacts to pollution has encouraged reevaluation of this practice. In this system, forage plants provide soil cover and contribute soil organic matter, while animal waste from grazers enhances recycling of nutrients and provides additional income to farmers when crops are not present. Manure in the forage plant residues allows for the slower release of nutrients compared to most nitrogen fertilizers, making them more efficient sources with much less loss to the environment. There are still risks of manure getting caught up in runoff, but the amount of nitrogen in manure is not as high as that in fertilizers, which lessens the potential for nitrogen to enter surface waters. Additionally, the use of other regenerative agricultural practices like reduced tillage, crop rotation, and cover cropping also lessen surface runoff. While there is a lot more that can be said about nitrogen cycling in agriculture, I hope this overview of the nitrogen cycle and how regenerative agriculture can play a role in reintroducing nitrogen into the soil using manure has been helpful towards understanding some of the motivations behind these proposed changes. In the next video in this small series, we'll be discussing the critical role of carbon to important processes in the soil, specifically in agricultural soils.
So that is, those are two of the videos. There are two more that are still not quite ready, and I didn't really have the time to share them with you guys today, but I hope that you guys like those. It's a little different than what we normally talk about in seminar, but I am going to get a little bit more into some research stuff now. So those of you guys that come for the data, here we go. So in addition to my video project, I also did a small research study in the lab, and I did a plant residue incubation study. And the purpose of the study was to conduct a 5-week incubation experiment to compare the varying influence of different plant residues on microbial respiration and mineralization. So this is sort of playing into more of the research side of some of the stuff I'm trying to talk about in my videos, with how like plant residues and different agricultural practices can help with soil health. We did expect to see some variation in the mineralization rate across the different residues that were being used. And in order to measure these different variations, I did use sodium hydroxide traps to trap CO2 that was released from microbial respiration. So you can kind of see those, you can see the centrifuge tubes that had sodium hydroxide in them in the photo. And I also did nitrate and ammonium assays to measure mineralization of organic nitrogen throughout the course of the study. So for my experimental design, I had 6 treatments, one of them being a control, so just soil. And then the residues I was comparing were soybean, sorghum, corn, cotton, and wheat residues. And like I said, to get CO2 measurements, I did titrations of sodium hydroxide traps, and I had 7 different time points. I did a 24-hour and a 3-day reading to get that initial CO2 burst. And then I also did basically weekly So out to 5 weeks total of runtime for this particular study. The nitrate and ammonium had the same time points as the carbon dioxide. And I used a KCl extraction to do nitrate and ammonium assays to determine ammonium and the total inorganic nitrogen reading. And then I was able to back calculate nitrate data. So before we get into the actual data, I wanted to talk a little bit about how I determined the application rates. So the application rates were equalized using total carbon of the residues. So as you can see, as you can see in my diagram here, I have grams of carbon per 100 grams of residue. And then I used the measurement for corn because it was roughly in the middle to determine, to equalize the amount of carbon so that even though I might've been adding more residue, is the same amount of carbon across the board. And so because of that, I actually ended up adding almost twice as much soy. So you can see there it's 4.08 grams per 50 grams of soil, as opposed to the others, which are more in the ballpark of 2 grams. And then I also have the carbon nitrogen ratios as well. Partially because I did notice the wheat had a lower carbon nitrogen ratio than is typically seen in the literature. And both of those things do end up coming up in my results. So speaking of results, I have my carbon dioxide data. So I did, you can kind of see pretty good curves here for CO2 accumulation throughout the course of the study. The blue on the bottom is the control. And then you can see this orange one here is the soy, which, like I said, was applied at about two times the amount of some of the others. And then right underneath it, the green is the wheat. And something to note here, it does peter off a little bit faster than the other ones do. And then I did run statistics on this, and there is a significant difference between the different treatments. And then you can also see there's some significant differences amongst these different treatments as well. So like between soy and wheat, not so much earlier on, but definitely when we hit that 2-week mark, so the 14-day reading, you can definitely see them start splitting up a bit. So kind of what conclusions I was able to draw a little bit. So when I looked at the soy residue treatments, they showed a higher rate of microbial respiration. This could be due to the amount of soy that was added compared to the other residues. And then with the wheat residue treatments, they did perform second to soy, but did drop off a little bit sooner than the other residues, which could have something to do with the higher nitrogen content or otherwise the lower carbon nitrogen ratio. And then for my ammonium data, this one, there was also significant difference here as well. And you can see the green, oh, I'm sorry, that is orange. The orange spike here is the soy, and then this gray spike here is sorghum. And there is some, we've got the gray is sorghum, and then we also have a later spike is green, and that would be the wheat over here. So it's kind of interesting. I don't have the nitrate data incorporated in here as well. It wasn't quite ready for presentation. I wouldn't really have enough time, but I do think it's interesting to see sort of the spikes at the 14-day mark, and then later on with the 28-day mark with the wheat. So some conclusions, in general, I was able to sort of determine that the amount and type of residue added can affect the rate of mineralization. So for the soy and the wheat treatments, they did have higher mineralization rates. The soy, likely because it was applied at a higher rate, and then the wheat because it could likely because of a lower carbon nitrogen ratio. And I did look into the literature a bit on this and noted that some possible causes, according to some other studies that are similarly designed, was like the lignin biochemistry can change decomposition behaviors. So like in the case of the sorghum spiking with the ammonium, that could be because of lignin content. And then also there were also some incubation studies that looked at like soy soy and other crops that showed that lower carbon nitrogen ratios mineralize faster than higher ones. So I think there's a lot of room for further study in this. You know, this is a small study that's hopefully meant to inform maybe a larger one later on. I think it would be interesting to do more incubations comparing, you know, the amount of residue applied like in the case of the soy, or similar residue types with different carbon nitrogen ratios. So like a low carbon nitrogen ratio wheat versus a high one, as well as run the study for a bit longer. So I did 5 weeks, and the ammonium was decreasing more towards the end, but the CO2 was still kind of going strong. So I think it could have been run a bit longer, and you might see something interesting as well. I also think it would be interesting to run and compare different residues than the ones that I did as well. And there is room for potential method development. I did this sort of old school with the titration of sodium hydroxide. I think there's a possibility of using gas chromatography to get more of a well-rounded view of greenhouse gas emissions as well, if that's something that you're interested in looking at. So kind of to tie in, the videos and the research and sort of how I'm trying to approach all of this. I think it's important to collaborative problem solving efforts across disciplines to find ways to disseminate information from what you do in the lab, what you do in the field, to other areas. And there are many fields that conduct a high volume of research that may never actually enter the mainstream. And it's not because it's not important. A lot of it is very valuable. It's more just because of the way the information, sometimes there needs to be ways of getting that information out to the public space. And there are many disciplines that have that. It's mostly an issue in public health, but I think it could be really helpful for climate change efforts and policy decision-making to figure out a way to disseminate information and keep it engaging and sort of get it into the public sphere where it can be used. So on that note, I'd like to thank my advisor, Dr. Howe, and my committee members, Dr. Smith and Dr. Liu, along with the Department of Soil and Crop Sciences and my fellow grad students and student workers. It's been awesome, and I really feel like I've grown a lot here. And yeah, on that note, if you have any comments or questions, I'm ready for those.
Great job. Great job. The music from the movies made me feel happy. So I like that. Questions?
Yeah, I have one question. I'm Shrudeep. So there is a great presentation. So I want to know what type of plant tissue you have used in this experiment.
What type of
plant tissues?
I used the residues left over in the field. So for the soy, sorghum, cotton, wheat, and corn.
Okay. So they're dry leaves or something like after the harvest?
Yes. Yeah. They were dried residues after harvest. Yes.
Okay. Okay. Thank you.
Yep.
Other questions?
I was just wondering, I don't know if you said this, but where are y'all going to post the videos? Are they just extension education resources? Are they part of classes?
Yeah, so some of that is still being worked out, but they're definitely going to be available for extension use. And then pretty much, I think AgriLife Communications is interested, maybe Soil Health Institute. There's a couple places, as well as for use in education as well. So it's supposed to be pretty broad spectrum, so it can be used in a number of different mediums.
Other questions?
Samantha.
Yes.
This is Megan Rooney. I think one of the things that you guys might want to look at too is reaching out to like the 4-H community with some of this information, because it's a great way to pull young kids that are involved in agriculture into soil science. I mean, I haven't seen any curriculum related to soil science for the 4-H kids. I mean, there's a lot of food and nutrition and ag economics, but I think your videos would be a perfect audience.
Thank you. Yeah, I'll definitely look into that. I think it could be good there too.
So Samantha, this is Larry Redmond, and I work in Extension, and I would encourage you to think about a career in Extension education, because we can use more of you that has the ability to explain these, what can be somewhat complex things, but you have the ability to break that down to where a lot of growers and youth could understand that. So thank you for devoting your time and energies towards this.
Thank you. I appreciate it.
Hey, Sam. So my question for you is, do you think that the current proposed soil health metrics are an accurate representation of what we should be evaluating, or do you think there's anything missing from those metrics?
That's a really good question, because I did look a little bit into what's currently out there when I was developing the videos, in terms of what's already been done for like extension. And I think we focus a lot on the science and how it impacts the soil. And I think that is extremely important. We are soil scientists, but I think there also could be some room for explaining more about how that affects developing communities. I'm very interested in working with developing countries and agricultural policies. And I think it would be interesting to look at the science as to how that affects like food shortage and that kind of thing in the long term. So there's definitely some room for growth, but I think we're on a good track.